Good morning. So this is a little different, mostly covered our research, our laboratory work. One of the student, Mr. Dr. Jayendar, who has done all this work, and I kept the photo, the work always I dedicate this work to my PhD supervisor Prasanna Mohanty from JNU, Jawaharlal Nehru University, who is no more, but I know. So I'm trying to walk into his shoes, but I hope I did, but still lot more to do. Okay, so here how the chloroplast behaves, or how chloroplast getting exposed to any drought, the stress is called drought stress, and we used a model plant, the Pisum Sativum. You know that Pisum Sativum, chickpea also we call it as. And this is a little bit different. Here is, can they keep memory? So when the stress is, when the plants are, even in an animal system. So when you expose, let's say somebody is affected with cancer. So even if treatment is there, still that the side effects will be remaining. Similarly, in the on plants, most of the research is derived from the plants because the mechanism is the same. So, but now I'm giving an example from animal to plant, plant to animal. You can. So now if you see the climate change that I think Professor Tripathi has explained the various climatic changes. The plants or animals, they get affected with the the cycle. So now, what is the drought does? Drought means less water, less rains. So now the mostly affected, the first affected people are mostly farmers because they are the one who always in the farms and looking for most of the dry lands, they with rain-fed crops. If there is no rain, they will be looking like that, waiting for the rain. And people are walking on the fields. These are I think rice fields, or maybe wheat field. So, and you can see all these cracks, and these are the can affect the land structure. The soil, the composition also it varies. If it is, that's most of the time in Hyderabad, there are many of the Indian scenario, when it's summer it comes, most of the plants die. Your garden, whatever garden you grow, all of them, having troubles. So if you look at the scenario of, another 20, 40, maybe, another 15 years, even India is going to affect with, dry land. You can see here the red marks. So all these red, and most of the countries, almost 54% of the agricultural lands is going to be affected by the drought. Okay. So now, what are the possible mechanisms in plants under drought stress? I mean, these are all known. That what happens if the drought is affected by the plants. So now, what the first one is, when the drought is affected, or drought stress reduces the water use availability because there is no water. You can see here water, and the signaling hormone ABA signaling will be increased. I mean, these these are all, and stomatal closure. The stomata get closer in order to protect our transmission, transpiration to not to evaporate water. Diminished CO2 flux, that means CO2 efflux will be reduced, and all these photosynthetically get affected, and the ROS, reactive oxygen species, increases to protect the plant, and a lot of see the attack on photosystems, and obstructed the synthesis of ATP. That means energy is will not synthesize, and if you go into the lower water potential will be, affect the plant, lower ribosco activity. I mean, if ribosco is activity is reduced, and obviously carbohydrate gets less carbohydrates, and leads to many other, other biomolecules also will not synthesize. And you can also see all these one, photosystem 1 and photosystem 2 decrease in, water use efficiency, there is non-photochemical quenching, and some of the proteins, they will be overexpressed, non-photochemical quenching, and PSBS is again a stress related protein will be expressed to protect the plant. This is a protective system. And also there is a stress regulating, the proteins or genes, the expression of LIA, genes and transporter genes will be, they express to in order to protect the plants. Okay, so ultimately what it does, the less of, the photosynthesis will be reduced. So we have taken here is a P is a model. You all know that, all of us know now the season now, it comes now P, you get nice P's. Now because there is a less research is, happening on, the P connecting to the drought stress. So, it contain, all of you know that, it rich in protein, sugars, carbohydrates, and, we wanted to see, I mean, there are a lot of, nutritional value and a lot of medicinal values, one could consume, we all get all this one. So, I mean, these are the things I am going to cover. And what Jayendra did here is, he induced natural drought stress. So, not watering the plants for, I mean, after growing, I mean, we cannot, if you start with, if you don't add water, there won't be any seed germination. Once the seed germination has happened after 14 days, he has given 8 days a drought, then again, wanted to check whether it is going to be recover the plants. So then again 7 days will be, they watered again, re-watering. So then we test all this one, the photosynthesis, biochemical, molecular, omic studies, entire, this project was, we have studied extensively entire, we have addressed most of the, the biochemical, molecular, physiology, biophysics, all of them we have addressed. We have published a couple of papers in this topic. So, very interestingly here, now 8 days when you give plants, there is a plants get wilted, I mean the leaves get wilted, and then after 7 days, so we keep one control. That means we don't induce any stress. We follow all regular watering. The other one we follow this one. The 7 day, why we have used 7 days? After 7 days it get flower. So there won't, we cannot continue. That is the way. That's why we cannot use. But the recovery rate is not much. Okay. Anyway, so now what we did here, I'm not going to. I mean, this you all know that. I think this is the, you have measured already this one. It tells about the photochemistry of photosystem 2. There is a OJ IP curves. Each component will tell about the photosynthetic water oxidation and the PQ pole reduction, and sending these one electrons to photosystem 1. So the data what it suggests here, this Fe bar Fm is a total photochemical reaction of photosystem 2, where you can see the day 0, this is the induction of the drought, day 2, day 4, day 6, you see that not much change. So when the day 8, I mean that after 2 days, there is a significant change in photochemical. This change is very significant, 0.85 to something like 6.1, about 1 difference, 0.1 difference is a significant difference. Then after watering, re-watering, it is reaching but not up to the control level. So then these are all talk about these two water oxidation complexes. Obviously, water oxidation is nothing but a splitting of water. So when there is no water, obviously the water splitting process get affected, where this increase is nothing but when you see here 100%. Now we use, these are the internal control, any in the protein chemistry. So if you don't have histone, histone you can use it, but still we use like 50, 100%. Let's say if you load one micromolar, 1 microliter or 1 microgram, and you use 0.5, then you use 0.25 grams, whatever milligrams or micrograms. So that is the dilution. It's called dilution. When you see here drought, there is a PSVO, one of the subunit, and PSBP, one of the subunit. There is about 50% has been decreased, which is an indication that the water oxidation complex is getting affected. Not only that one, we also looking at this one, we can tell many other things. There is a disturbance, or disturbance between the pigment to pigment and pigment to protein, that we will discuss a little bit later. So similarly, whatever the handy-P data, we can also, there is a PAM instrument, it also tells about electron transport. This is the electron transport and the yield of photosystem too. So this tells about that there is a decrease, significant decrease at the day 8. You can see very significant decrease is there. And similarly, the non-photochemical quenching is nothing but a excess energy is quenched. The excess energy, that is very important. Plants get protected. When you, let's say, all of you, if it is too much summer, okay, especially the average of Indian summer will be about 35 to 40 degrees. So you cannot stay in the summer, 10-15 minutes. Can you stay in 15 minutes in summer light? You cannot. What you do, you go to shade. But plant doesn't have that mechanism. They cannot go to shade. They cannot walk. So what they do, that anything light is falling on the leaf, they have a mechanism they have developed that is called non-photochemical quenching. That excess energy, whatever, I take only this much water, let's take a water or whatever. After that, I reject, right? When you feel your stomach is feeling hungry, you take only the amount which you require. Even if you keep a food, bowl, big bowls, or we have mandi. Today is going to get mandi. So if you put mandi in front of you, you cannot eat entire mandi. So you eat only the 10% or to 15%. But the same thing, what it does here, the cyclic, the non-photochemical quenching is around here actually. So this place, and these are the two very important subunits, that PSBS play a major role in increasing the the delta pH, that is called hydrogen ion protons. The protons get increased, and this balances the cyclic electron transport. This switch now from linear to cyclic electron transport during the non-photochemical quenching. Is it clear for you? Okay. Okay. Then maybe photosystem 1 also getting affected. I don't want to go into more. So what it does, so why they are doing it? So the question is why there is change in photosystem, why? Mainly you all know that the main culprit is ROS, reactive oxygen species. So what it does, there are these are the reactive oxygen species generated by the plants, singlet toxin, peroxides, and hydroxyl radicals and H2O2. So these radicals are mostly affected the plant. The plants get affected if they generate all these one. This is called oxidative stress signals. These signals. So whether all of them are producing by the the drought stress, but that's what we'll see here. So what we have done all these superoxides, H2O2, this APX, catalase, and SOD. You can see here a significant increase, many folds of increase of all these is an indication that these are all playing a major role in protecting against the doubt, but the enzymes are there to scavenge the reactive oxygen species, but still plants are able to survive. That's what I said. Plants are able to survive. One is non-photochemical quenching. There is other one reactive oxygen. There are many other things we'll discuss. Okay, so now the same thing we showed using Western blotting. Western blotting is nothing but antibodies. We against these proteins we have antibodies. So we tested the significant increase of catalase, manganese SOD, APX, and copper zinc SOD. You can see this in 100% drought. They have a significant increase. We showed this one in the relative increase in the graph. So what it does, this one of the one, we can read this one. There is a decrease in photosynthesis, and because the increase of ROS and non-photochemical quenching. That is the summary of this one. So now, so I said oxidative stress appears to be reason for induced and disassembly and aggregation of photosystems. So the proteins get aggregated. That's what we're going to discuss now. So this is, I think you are done yesterday, this experiment? Already loaded? Okay, we are going to load today. So now I mean, just let me know, they might have explained Pavitra and other one. So the thylac, I mean, anywhere in a Chlamydomonas or the algal system, or the cyanobacteria, or the higher plants, they have this, and Professor Tripathi has explained very well, stroma lamella, grana core, granum margens, all of them. So in which inside in thylakoids you have a complexes like photosystem 1, photosystem 2, light arresting complexes, photosystem 2 dimers, photosystem 2 monomers, and LHC2 trimers and LHC2 monomers, and ATP synthase complexes. All these one can be separated using the blue native gel electrophoresis, which Pavitra is explained yesterday, right? Pavitra, you have explained? Explain is not done. Okay, now this is another one. We can see here. So these are the complexes we could able to separate from the blue native gel. We isolate the thylakoids. Thylakoids, right? Chloroplast. You disturb the chloroplast, you get thylakoids with using differential centrifugation. After that, we use they're going to explain about the beta-DM, the detergent, which the detergents will be one. It kind of it makes a micelles inside micelles. The micelles will create loosening of the complexes, and through electron electrophoretic mobility, you can separate all these complexes. These are blue native gel, similar to SDS phase. It's also called a native pair. So you don't add here denaturing agent, beta-mercaptoethanol. Without that, so you can separate these complexes. So all these complexes, PS1 NDH complex, PS2 LH2 super complex, big complexes, PS1 dimers, I said, ATP synthase, PS monomers, cytochrome B6F complex. Whatever you see the thylakoid, the composition, schematic representation, which I showed you last class, all this can be separated using this one. You see, I told you trimer, dimer, monomers. So that's what we are going to see. What happens to these complexes under drought stress, which you can see later, the next slide. So this is the one. So drought, control, and recovery. The thylakoids, are separated using beta-DM, but electrophoretic mobility. All these complexes. So I mean, drought also, most of the complexes are almost intact, but there are some differences, especially the dimer, LH2 trimer. Dimer is little increased. I mean, this is only Comesis time. Both are same. To see much clarity, we have done the Comesis. And there are some, especially the super complexes. The complexes, they have super complex means bigger complex, larger complexes got affected by the drought. So that is that because of that, you see whatever the change in the photosynthetic activity. So because change in these, they become looser, right? So like a spring, you do it like that. If you keep like that, that means they will be stretching out. That stretching is the one you could see here. When it's stretching, this is a spring structure. I mean, it comes back later. Once you put water, it comes back to the regular natural conditions. You can see this one. So that is what we could see. The drought is inducing the change in supercomplexes, this one. So then further what we did, I mean, there is also, you can see that one, the trimeric complex also little bit increased compared to control. And when coming back, they change these complexes, which you can also see here. So I said, aggregation. The proteins are getting aggregated. So now, what to show that one? I mean, we have done all these very interesting, to understand the aggregation. We solubilize the thylakoids or anything. You can apply this one to any research, not only the chloroplast, even in many other studies. So like, this can be like any disease, even human diseases also. So you just, if there is anything membrane related complexes, so if you want to understand what are the markers to markers against specific disease, to probe that one, you have to go with, the proteins, otherwise, you go with proteomic data or whatever. So what we did, these solubilized fractions, again we run the SDS page, then probe with the antibody, and where you can see here, these are some of the PSA, B, and and also the CP43, 47, LHC-B2, LHC-B5, and all these blue color has enhanced into drought conditions. When you see this one, 100%, 100%, you can see here, they significantly increased. That means when the solubilized fraction and the precipitate in the down, when you get centrifuge, in the down, in the soluble one and down, you will have a precipitated. That precipitated is nothing but aggregations. Proteins get aggregated, tend to aggregate if there is any kind of stress. It can be applied to plant or animal or any kind of these human samples also. So these proteins are playing a major role in aggregation form. Why they have aggregated? That is again a story that because of ROS, they are getting aggregated. So we did use that one, using confocal microscopy, where you can see this is the control, and this is the one, the treated, that means drought. You can see the clumps, very big clumps. I mean, the different magnification. You can see a very red spots where you cannot see much, but this is an indication. But this again we have run the SDS page to see this one. What are the proteins are getting aggregated? So what we said, these are the proteins are getting aggregated. So hence we showed other the green gels, and where in the reverse recovery you don't see much in this one. That means, and it comes back to the plants are always, I say that they're a beautiful mechanism. Like not an animal, like plants, even if the leaves are falling, pour water, again regenerate and comes back to natural environment conditions. But not always. But if it is a disease like pathological effects, very difficult. But abiotic stress, they can come back to original conditions. Okay, so we showed again we using a circular dichroism. We are not measured it, may not yet. Today, no? Okay. So this is the one, you can say that pigment protein complexes can be understand pigment interactions, and pigment protein interaction also can be understand by using circular dichroism spectroscopy, where we showed a lot of significant change in the pigment composition. Pigment, you see that in the drought, it significantly affected this region, that is a mostly chlorophyll A region. And similarly, there is a carotenoids also getting affected. This is the carotenoid region, this is also getting affected. So there is a change in chlorophenol and change in chlorophylls. So this is again, I think Professor Tripathi was talking about this the structures, this electron microscope structure, you can see nice, now this is the control and this is the drought and this one is the recovery. So, where let us take this one is the same magnification and this one same magnification, this one same magnification, this is same magnification and same magnification. So, and you can see here this size, okay, this size and this size has significantly increased, that means it's kind of a swollen. And also you say that you can also become, like because of there is a pigment interaction has been changed and pigment protein also has been changed. So, that is why it is becoming loosening. The stacking is whatever they said yesterday was nicely explained to you, right. So, there is becoming because electrostatic interaction is there is change in electrostatic interaction, they become loosen because the ion mobility has changed. So, that is why because the drought there is more of water is going out. So, there is a change in the mobility of ions. So, there you can see the lot of change in this one, it is increased. And whereas in reversible, I mean this comes back to the native state. So, similarly, what we did just created a model. I mean this is the model where you see that Gainer stacks the repeated distance. So, this is the one I said increased, those repeated distances you can see here it is increased in the drought stress. That is again I said it is because of less of water and many other things, many factors that we have showed. Maybe you can share the paper, you can going through that. Okay, I am not going to discuss this one because already Ichiro Takahashi is explained, and now we wanted to see what is happening to the photosystem 1. So, on photosystem 1 and photosystem 2, the structures, these are the structural the components, the dimension, and these are the photosystem 2 structure compounds and their proteins, and we showed all of them using Western blotting. We probed all this one, and you can see their change when it is expected to drought and how they have changed. You can see here some of the proteins like CP43, and also the PSBO, PSBB already showed you, and there are also like like the LHC2B, that means photosystem 2 harvesting, light harvesting complex also you can see here significantly changed, and maybe LHC5 also changed, LHC6 also changed, and as I said dehydrin and aquaporins are the one markers for drought. Reviewers will ask what you have used, what now you have induced drought, what is the evidence, how do you prove that this is affected to the drought. So, to show that one, the aquaporins and dehydrin is the one you see here a very significant increase. There is no dehydrin is nothing but hydration, I mean dehydration. So, when there is no water, these proteins get overexpressed, and you can see here very significant increase in the subunit. And similarly, aquaporins also will be have increased here. And now similarly photosystem 1, there are some proteins PSA and some little bit PSB and some of the proteins like PSAF and some they have reduced. You can see especially these two PSA B and the F they changed, and mostly LHC1 is almost like LHCA2 is reduced significantly, and LHCA4 is little bit reduced, but otherwise all of them are stable. These are the light arresting complex of photosystem 1 they change. So, okay, to conclude to this objective here, you can see here in all these biochemical, biophysical, the NPQ is induced, and some of the specific proteins are getting overexpressed or during aggregation process, and these I think Lea dehydrin proteins are expressed, and some of the light arresting photosystem 2, photosystem 1 proteins get aggregated and drought says. So, yeah. So, now this you can check this paper, if you want to understand more, we have given, all the details in this paper. Okay, now this Ichiro Takahashi is explaining about the complexes. What is happening inside the complexes? We have separated them using sucrose density gradient. You can see these complexes, F1, F2, F3, F4 is, there are fractions. So F1 is light arresting complex 2, F3 is PS2, F2 in F3, you can see here, F1 LHC2, F2 PS2 core dimer, and F3 PS1 LHC complexes, and last one is, F4 is PS1 LHC1 complexes. So I don't want to go into this one, I will skip this one. Only what I want to tell you here, the pigments, there are, and just to conclude this one, I don't want to go into more into this one. So what, from this sucrose density gradient, what we said here is, there are different, you have seen the fractions, LHC2, PS2 core dimer, and PS1 and PS2 and light arresting complexes, and last one is PS1 LHC1 complexes. And these complexes, they express, different carotenoid pigments and chlorophyll pigments. And there are differential change in the pigments, which leads the change in the, protein and pigment complexes, change in the pigment. That's what we wanted to say. If you want to go through it, we can go through our laboratory work and, lot of publish. So now that, and we also checked, using RT-PCR or Western blotting, reconfirm whether whatever we have done is correct. So we used ZEP means zeaxanthin, vilozanthin, all these one you can see in the drought condition, they have significantly increased, which is we can say that a marker for identification of non-photochemical quenching, and also the increase in the carotenoid biosynthesis during drought conditions. And we did same thing, we have tested all these, the genes by using the RT-PCR, where you can see that most of these pigments, that means, let's say beta carotene to zeaxanthin, you can see the CHYB, you can see the increased here, and and if you can see this in some of the genes, xanthophyll cycle, and the CHYB, you can see here is increased, and PDS is mostly the conversion from this one, phytin to lycopene itself is increased significantly, which is an indication that the whatever, the pigments coming from, the chlorophylls are converting to carotenoids, and these are the one, all these compounds have increased. Okay, so now, this work was, we can also share this paper, the previous one we published in Plant Cell Physiology, and this work is now, it's also in the web, recently published another good paper, 3 good papers. It's a plant cell environment, there is another paper. We showed all these one, what is the connection between lipids, membrane lipids, how the membrane lipids are playing a role in photoprotection against drought stress. So if you look at this one, possible mechanism plants under drought stress and polar and membrane lipids, you all know that membrane lipids, we have 4 membrane lipids, MGDG, DGDG, SQDG, and PG. So these are 4 major membrane lipids. So when the plants are affected to drought, so there will be a production of ROS, all this we discussed. They can affect the DNA, protein, lipids, and then the lipids, they will change the lipids like MGDG, DGDG, SQDG, PG, and in the PI, there are some plants. So what they do, always the lipids, when they convert into different forms. Why they convert? Any of you can tell? Any lipids like a carbohydrates also, they convert into one form to another form. These are metabolic conversions. So this is mainly because to produce ATP, the energy, or this is a protection mechanism, so that if this is the lipids are converted to triacyl glycerol, there will be a lot of energy generated. This energy, or this also what maybe this because the directly the ROS is getting affected to the lipids. So now another form they trying to accumulate. So this is the accumulation is triacyl glycerol. They accumulate, and when it plant is required, let's say a plant is getting stressed, let's say too much stress, the lot of triglycerol is accumulated. After the accumulation, what it does, when the plant you water it again, these guys can convert into again lipids to the MGD. Similarly, this is one way to the lipid getting affected. Then another one, attack on photosynthetic membranes, where you could have seen that first two objectives which I told you that all is getting affected. So these are all the antioxidant protectant. There are osmoprotectants. There are antioxidant mechanism. They're all one the stress defendant. Stress defense mechanism, SOD, APX, which I showed in the first chapter. First, the objective proline, they will be all involved. They increase during the stress condition to protect the all the plants or animal, even an animal system also. The same kind. The mechanism is same in animal system. Okay, so what we did here, just want to show that, in the drought conditions, this is biomass, we already, less biomass, and carbohydrate content is increased, is significant. It has to increase because there is a, the alternatively now it accumulates more carbohydrates, and starch content, since carbohydrates, starch also increases, and the sugars, these we have identified sugars, and proline, you see that a high amount of proline. I don't know how many folds, 5 to 30 means 6, 5, 6, 7 folds more, and leaf area is reduced. Obviously, leaf area when it's getting affected is reduced. So we also tested other, they have increased significantly. So then what we did, we separated the membrane complexes or membrane lipids using TLC, thin layer chromatography. So after separation, we have separated all these the lipids, MGDG, DGD, SQDG, and TGDS, trimethyl homoserine. Sometimes we get, sometimes depending on the plant, and phosphatidyl glycerol. So they looks like that, Then you can see here. First thing what we did, the triacyl glycerol separation. One is, the triacyl. This is basically organic solvent. Different organic solvents matters. So you can see here in drought stress, MGDG is reduced when you see this one, and DGDG and PG, they all that means they have converted. They have degraded actually. So this degraded, and this is coming back to the control level, but not up to the one in the recovery. So and SQDG also changed. So what we can see here, when you see these in TAG, that's what we wanted to show here. And they have increased. This is the these two are the TAG, that means triacyl glycerols. In control, not much. They have increased slightly. This is indication that they are converting into these. They convert into TAG. Maybe not all of them, but some part. Okay, now we did this one to 2D. The one dimension, second dimension TLC, where you can see here, the drought, MGDG, there is a change, the DGDG is significantly changed, and the SQDG, you can see that very, almost more than 60% has changed, and it is reversing in recovery conditions. So we did all this one fatty acid analysis and showed, you can see here, some of the control, recovery, for 42% you can drought is significantly increased the fatty acid composition. That again, when the fatty acid is degrading, that means beta oxidation happens, there will be lot of ATP molecules, the energy generated, that energy is utilized by the plant to cope up to the plant, to the stress. And you can see that significant increase in saturated fatty acids. So then this is the outcome of this one. The conversion of these, all these one, I said acetyl CoA is the one. Through acetyl CoA, you get all these, the final products. So these PGAT and DGAT, these are the one. The conversion of membrane complexes, it comes somewhere, here. They convert into acetyl CoA, then it comes here, and here, that, and from there, before the generating a triacyl glycerol, the PGAT and DGAT are the one playing major role to involve in conversion of the TAG. Where we wanted to show here, you can see these 100% here, compared to here, almost more than twofold has increased. This one and this one. Similarly, you can see here this one and this one. And also we showed something, ATG is also increased, autophagy is, that is a different story, and I am not going to discuss. Autophagy also is significantly increased in drought stress. So the meaning of this one here is the lipids, membrane complexes are, not only membrane, any other lipids also is converted to TAG, apart from membrane complexes. This one, the one we have seen, polyunsaturated fatty acid. Polyunsaturated fatty acids are significantly increased. These polyunsaturated fatty acids are involved in conversion of TAG, where they are coming, where polyunsaturated lipids are coming, they are coming from membrane complex, membrane lipids. And other, DAG also is increased because maybe some other lipids also coming. Not only the membrane, maybe other lipids also converting into TAG. Okay, to conclude this one, these are the, the conclusions. The TAG accumulation again coming from membrane complexes. There are a lot of significantly increased the osmolytes like proline and even a lot of glycerols they have accumulated. Okay, so now little bit about omic studies. Too much of you have I will not talk about all this methodology. We did a lot of omics, like all omics, genomics, transcriptome, proteome, metabolome. We connected all of them and understand, and to understand the markers, and these markers now will be using to develop the against drought. Now we'll be using, we'll be using that one in many, even chlamydomonas. You can use those markers to develop stress resistant plants or stress resistant algal system. So I will not go into deep into all these analysis, I will just only go with some of the markers where we have, these are the upregulation of transcription factors. What we have seen, there are the ABA is one of the major one, and some of the heat shock proteins, and we will also see that these are the drought, you see that control and drought treated, where you can see these genes, PSB and PSBS, which in Western blot also we showed some of the the markers, they have increased significantly, and now these are again the markers, they are significantly increased. We want, and some of the photosynthetic pigment, these are very, you cannot read them. Some of the photosynthetic genes also been significantly downregulated and significantly upregulated, some few of the photosynthetic proteins. Then we did photosynthetic genes or transcripts. So now we did the proteomic analysis using the ITRAC method, where the samples was analyzed by UC Berkeley. From we sent the samples to US, they are given the data. So where I will not discuss all of them, because to show to the genes, and what are the genes, the same genes we have analyzed with the proteins, and you can see all these one is similar to the gene expression or transcriptal expression. The proteins also getting similarly getting overexpressed in some of the places, and you can also similarly we tested with the transcriptome and the analyzed together. Some of the, see the photosynthetic protein, these are all photosynthetic proteins, PSAB, that means photosystem 1, photosystem 1D, photosystem 1E, photosystem 1F, They are all significantly decreased, which is agreement with the other biochemical data. Okay, this, I mean, to show this one, I mean, this I already showed, but to show again, this is the Western blotting data, which is matching with the proteomic data. So then again, we tested many other, these are all the, again, probably you might be knowing all these one, DNAJ, thyroidexin, COX, NOD, nitric oxide related genes. They play major role in stress regulated mechanisms. So what we are now working and trying to understand, these genes, can we improve the plants, or can we improve the algal system against a stress condition? But they are very specific to drought. There are also some other genes. This is nothing to do with protein. There are the photosynthesis. There are some other. They can also see here some of the genes of the thyroidexin, iron sulfur cluster related cytochrome 450. They are also increased significantly protein side. So we did a lot of network analysis, which I'm, network analysis will tell about whether whatever the proteins are, the genes, how much interaction is there from one protein to another protein, which is, you guys maybe know much better than me. Okay, we did all the metabolomic analysis. We have identified all these one. I mean, I'm not telling that we have identified 35 metabolites and 3 groups. All these groups were significantly increased in drought stress. You can see this in carbohydrates, polyols, and other metabolites. So you can play around these metabolic, and improve, or let's say they have increased all these one. Maybe you can, the genes of these specific metabolites, you can also manipulate. That means you can knock out, or you can see that mostly you do that knockout, and the biggest is more overexpressed, maybe silence it, then see that you can able to protect, or since I don't know, there was yesterday a beautiful talk was given by somebody from US scientists. So the CRISPR is the one now telling multi-genes you can play around. So multi-genes, about 10 genes at a time, you can you can silence, or you can knock out. So let's say these are one specific genes, you silence it and see that if they can able to protect against stress. Maybe in future we'll be doing all this one. Okay, so to conclude all this one, you can read this here. Our transcript data revealed that most of the photosynthetic genes are down-regulated, and stress markers genes, especially dehydrin, PSBS, and sheath shock proteins, and aquaporins, and are these are increased, and majorly photosynthetic genes are down-regulated, because when you can see that photosynthesis getting affected, so where we could see all the most of the proteins are related to photosynthesis, they decrease. And the ABA responsive elements, ethylene responsive transcript factor were upregulated in droughts. That has to be there. If the ABA, the play hormonal effect is very important. And the same thing, proteins, both photohistone 1 and photohistone 2, the proteins getting affected, and RYS and the heat shock proteins enhance performance, leaf drought conditions, and some other proteins are also, they're kind of stable or increased. So all these one, the carbohydrate and polyols, so they have increased to maintain the cell integrity. So that's why always the these are osmolytes. You can also say the osmotic. They maintain the osmotic because of the carbohydrates and polyol. If they are not increased, and the plants die immediately. When you give a stress, they will get immediately affected. So overall, this is the summary of the entire study of the drought stress on chloroplast, how the chloroplast getting affected, chloroplast metabolites, proteome, the transcriptome, other physiology and biochemistry, biophysics, getting affected. You can clearly you can see that one, see that downwards is decreased and upwards are increased. You can take picture, and now we can also, anyway we will be sharing all this material with you. So with this I conclude, and if you have any questions, I'll be happy to answer. Any questions? I said these studies can be implemented not only plants and animals, especially the ROS mechanism and their protein aggregation, and the metabolome studies can be applicable to even humans or animal studies also. So likewise, only the discovery happens animal to plant. Many Nobel laureates, the autophagy Nobel has come from plant to animal. So they are tested, they have taken plant genes. Then the Japanese guy got Nobel Prize. The many other a lot of Nobel laureates all getting discoveries, reading more articles from plant side. At the same time, sometimes it also happens from animal to plant also. The recent studies like movement, plants also can speak, right? Have you studied that article? They communicate. So they also cry. Plants cry. Now that's what they prove that the studies are proving. There's a couple of Nature papers has come last 2 years back. Yes. Yeah, we use the markers. Basically, markers, known markers. Yeah, markers we have to give. That's what we use TAG, triacylglycerol marker. Otherwise, they're difficult. So then MGDG, DGDG, SQDG, and all you have to use markers. Markers, the literature. I mean, there is already a standard protocol. If it is MGDG, DGDG, SQDG, these, it is known, that, it's a separation. Based on literature, you can identify. But, we had a lot of difficulties publishing this one. They were keep asking us to show, I mean, we believe, but even if you cite literature, they say that, how can we believe that, your, whatever the bands are, when we have to do it. Actually, we didn't show here, but in the publication we showed using standards. Otherwise, when you submit to big journals, it's difficult. Now, especially these lipids, when you are running on TLC, you have to use standards. Without standards, now even if going to LCMS, it gives different. It gives only fatty acid, right? It won't give you lipids. Mostly fatty acid, right? So, but there is one more you can use it, FTIR. But FTR is only functional group, yes. No, you cannot get FTR also, but the lipids, based on that, there also it is qualitative only. Quantitative, I mean, to really identify, you need to use, without markers, difficult. But many people, they agree, with the TLC, when you run TLC, it's already known. MGDG separates like that, people know that. But like a crazy like me, I may, like if I'm reviewing, I want to reject the paper, I will ask, okay, you have to do it. So those are wise people, yes, okay, it's a known procedure, why to, bother them. So it's like that. But better to have the markers. My students also can ask questions. So, no questions. Okay.